Thank you, Ingolf. I have a very nice husband. He believes in a happy wife, and the smell of skunk helps. This morning, I want to talk about sensory descriptive analysis. And to get there, I want to talk a little bit about sensory evaluation, about human variability. Then I'm actually going to show some data, which may mean that it's going to be a little too technical. However, I hope that at the end I could be able to bring it back and show you what some of the things that you may want to do when you try to drink some wine or try to evaluate some wine. Okay, sensory evaluation is a science in which we use human beings to give us information about products. And if you've ever bought deodorant, this is how things happen. If you've ever bought Huggies, if you've ever bought a Nokia cell phone, if you drive a Renault, all of those saw a sensory scientist somewhere along the way. So what we do is we give somebody a product, we evoke the response, we measure what they tell us, we analyze it, and then we make decisions. It's pretty much straightforward science. However, we are using a measuring device that has issues. It's a wonderful device. It has a brain. It can tell us things. But on the other hand, it has a brain. As different as these people are on the outside, as different as all of us are in this room, our senses are as different, if not more so. Those of you who were there yesterday when Dr. Breslin talked about amylase copies and salivary amylase, you went from 2 to 12 for people. That's not unusual. That change in amylase copies actually changes the way you perceive the texture of starches when you consume them. Similarly, most of us, I think, have heard that there are some colorblind people in the world, somewhere around 0.5% of females, because you need to get it from a colorblind father and a mother who is also a carrier, and somewhere between 5 and 8% of men. However, there is also great variation in normal color vision. Not a one of us in this room probably sees red exactly the same way. Not a one of us in this room probably sees green in the same way. And it is likely also true for blue. The example I have here is one example. It's probably the most studied example. There are others. So in your red pigments, you have amino acids. That's how those receptors are made up. If in the 180th position, you have serine versus alanine, it changes the way you see red. If you have serine, you need less red light to make a match for yellow. And so the way they study this is by giving you a yellow target and then asking you to recreate that target. And if you need less light to match it, they can also then test your pigment amino acids, and there's a difference. The difference for green in the same position is not as dramatic in terms of the numbers of people, but that's one position. The thought is that there's probably 20 or 30 more where these changes change things. So we see the world quite differently. Supertasters, a word I absolutely detest. I wish we had called these hypertasters and hypotasters, because super seems to have a connotation of better than. In actual fact, it's different from. About 25% of us are supertasters, and about 25% of us are non-tasters or hypotasters. A little higher percentage of men in the American population, a little higher percentage of supertasters in Asian populations. These people get identified by being able to taste a compound called phenylthiourea, or PTC, phenylthiocarbamate, which is a carcinogen. Originally, this was used by anthropologists extensively, especially in Polynesia. We don't use that anymore. We do use probe, 6-normal-thiouresyl, which is a medication for goiters. The quantity you need to test a supertaster is significantly less than you would ever need to use for medical purposes. It's genetic. In other words, you can't change from a non-taster to a supertaster or vice versa. And it may have had an evolutionary advantage in that compounds that are high in the group that causes this change are toxic, and it also seems to have some protection against goiters. So supertasters tends to be less prone to goiters. Supertasters not only can taste probe very strongly, and we're talking about minuscule amounts and they're gagging, whereas non-tasters will sit there and you give them more and more and more. It's like, well, when's the water going to stop and I'm actually going to start tasting something? Supertasters have a totally different mouth environment. They have many more taste buds, which means they have many more taste papillae, because all of our taste buds are on papillae. Every one of those taste buds are innervated by the trigeminal nerve. It's a 3 nerve. It goes to the eyes, the nose, and the mouth, the mucous membranes and the mouth. It's what makes you cry when you cut up onions. It's what makes you sneeze with black pepper, and it's what makes your mouth burn when you taste capsaicin or chili peppers. So they have many more of these papillae, many more taste buds, many more pain receptors in the mouth. Another way you can identify a supertaster, and you can actually do this at home, is get some blue food dye, paint your tongue, then find a reinforcement circle. You know, those old-fashioned things we used to have. I don't really know where we use them anymore. But anyway, stick this thing on the tip of your tongue to the one side or the other of the midline of the tongue, and count the little red dots you see. If you have more than about 40 in that space, you are probably a supertaster. If you have to saddle up a horse to try to find a few, you are probably a non-taster. So usually less than about 20. Between 40 and 20, you're probably a normal taster.
How can you see that?
If you paint your tongue blue.
You need a second person, I bet.
Well, mirrors. They exist. The papillae won't stain. They will stay red. So you'll have these little red dots to count in this blue background. Our dean wants us to create a national flag for an exhibition in Milan that will do this. And too many national flags, well, ours included, have red on them. You can't use it. Okay. Why is this important when you're tasting wine? Supertasters tend to be not lovers of kale, for those of you who were there on Friday night. They tend to hate cruciferous vegetables. They find them very bitter. You can improve that situation by roasting said vegetables and adding sugar, or threatening them with death if they don't eat. But in the case of wine, they see alcohol very differently. They, first of all, see alcohol as being intensely bitter. This is a 10% solution of alcohol. So if you take a shot of vodka and you add 3 parts water to it, so we're not talking very high concentration, they find it intensely bitter. Non-tasters, yeah, it's a little bitter. They find it extremely hot or burning. Non-tasters, not really. So they have a very different mouth environment in general. They tend to have issues with textures, because remember they have all those pain receptors floating around. They tend to be horribly picky children. They can grow out of it. Not the super tasting part, but you can, we have a brain. You can choose to ignore the signals being sent and start eating with the family or whatever the situation is. Let's try another sense, aroma. Probably none of us live in the same smell world. This has not been very extensively studied. It was studied quite extensively in the 60s, kind of went away, and I would say in the last 5 years there's been a huge amount of work done on this. And that is specific anosmias. This means you have a good sense of smell. You don't really have any issues. But there are some compounds for which you are much less sensitive than the average, 100-fold less, at least. When Amor did this in the 60s, out of the 62 compounds he tested, he found 40 that had at least some percentage of specific anosmias. One example would be androstenone, bortaint. About 5% of the population is totally anosmic. And there's a receptor gene for it. If you have both copies of this gene, you found this compound very unpleasant, urinous, nauseating, disgusting. If you only have one copy, you're going to say, it kind of smells like gym socks or a locker room, or, yeah. If there's a slight mutation in that gene, it smells like flowers. Lucky you, we need to find more of those people. Here are a couple of compounds that are 30% anosmia, so at least 30+% of the population have a different odor response to it than the other part. Isoburaldehyde, which smells like wet cereal or straw, geraniol, which is a rose-like odor, glutaraldehyde, which is a pungent disinfectant, and musk odors, used extensively in male deodorants and perfumes and aftershaves. Well, probably 30+% of you have wives or girlfriends or others who can't smell this stuff. To bring this back to wine, here are two compounds that are found in wine. One is alpha-ionone, which smells like violets. The other is beta-ionone, which smells like roses. They are stereoisomers, so they are as similar as my two hands are. They're just opposite directions. In the case of alpha-ionone, we get a normal distribution. Most people smell somewhere in the middle here at about 100 parts per billion. There are a few people here who have to have huge quantities, 5,000 parts per billion, to smell this. And then there are a few people over here that are probably at about 1 part per trillion or so. So even in a normal distribution, there's a huge differentiation. Now we have this one where we have specific anosmias happening. We have this group over here who are extremely sensitive to this compound. They need essentially nothing. Parts per trillion, 1 part per trillion, 1 drop in an Olympic-sized swimming pool, and you're smelling in roses. You need nothing. There's a whole bunch of people here who smell this stuff very sensitively. And then there's a whole herd of people up here who really need a huge amount of it. Their wines are going to smell very different to each other, correct? And we can go on and on and on, and that's not the point of the exercise this morning. So, with all of this situation, how can I possibly use human beings and get anything that I can actually use? The answer is with great caution and care. There are two kinds of sensory evaluation, and I'm only going to talk about one this morning. The one I'm not going to talk about is consumer sensory evaluation, which is a subjective measurement of liking. It is hugely subjective. It is hugely variable. It is hugely variable across people, across cultures, in every which way you could possibly look at it. If you do it correctly, and if doing it correctly in many cases means using many people, and then using segmentation, you can actually get some really interesting information. That, however, is not what I want to talk about today. I want to talk about the other type of sensory evaluation, analytical sensory evaluation. In this case, it is not subjective. It is objective. We use trained panelists, and we are interested in the product, not in the liking. In the realm of analytical sensory evaluation, probably the most useful tool is something called sensory descriptive analysis. And in that tool, we use trained panelists, usually somewhere between 8 and 12. 8 being, from a statistical point of view, a good place for us to deal with some of this variation in people. 12 being an easy number to train. If you have a dinner party and you have 4 people around the table, you are likely to have 1 conversation. If you have 15 people around the table, you're likely to have 12 conversations. During training, you want a single conversation. You want everybody to pay attention. And so most panel trainers can handle 12 people. If you're really good, I had a graduate student a few years ago who could do 22 at a time. Beatrice was scary in a very gentle way, but she controlled 22 people and had a single conversation. You have to have extremely well-designed experiments. You have to do lots of replication, because we need to deal with all this variability stuff. Also the fact that people get up in the morning and they're grumpy or whatever their problem was that day. And so you have to deal with a lot of statistics. So here we are doing a descriptive analysis. In this particular study, we were looking at 17 high-end wines, $40 to $190. We had 22 judges. This is Beatrice's panel. We start out by having all of them smell the wines that they are going to be seeing and coming up with terms that describe differences among these wines. So if you have two wines that are the same red color, red is not a good term. But if one is lighter red than the other, then red is a good term. So they start out by coming up with, everybody comes up with their own idiosyncratic terms. You then write those terms down on a board. Everybody participates while this is happening. So somebody may say it smells like cis-rose oxide. That's probably a flavor chemist. Somebody else may say it smells like roses. Somebody else may say it smells like Gewürztraminer. All of those are the same odor. So you can start boiling things down, trying to come up with terms that are the same. And the key is to get everybody to have the same concept of that term. Because we use words, especially smell words, extremely idiosyncratically. We may have exactly the same term for two totally different things, or different terms for exactly the same thing. One of my first panels I trained, one person insisted it smelled like violets. The others insisted it smelled like canned blackberries. I got all the violet standards, which we'll talk about in a minute. This guy kept telling me, no, no, no. I was going insane. Among other things, I picked violets off the chancellor's front lawn. And in the end, I said, OK, we'll get back to your violet situation tomorrow. We'll deal with the blackberries. I handed him the canned blackberries. He said, violets. Which solved the problem right there. But you can also have two people say violets and not mean the same thing, or blackberries or whatever. So once you have all the terms, you then start creating reference standards. And we'll get back to reference standards in a moment for every single one of those terms. And these reference standards have to be approved by the panel. So I can't go off and get a banana and throw it in there and say, that's the banana standard. I have to bring them options of banana. And they have to tell me which of these actually means banana to them. We'll get back to those in a minute. Once we have all of that, we then start playing with these terms. So they have a list of terms here. They're trying to find out if they're really there. Do they want to change the references? So there's a lot of back and forth, probably about an hour a day for about 2 weeks or so. At the end of that period, they should be comfortable with this set of 24 descriptors. But you know, it's easy to say something smells like banana when it says banana on the glass. And you stick, oh yeah, that was banana. That's really nice. We then test them. Dr. Manning here is a retired microbiology professor who does a lot of panels for us and is a really good model. And he now has glasses with 3-digit codes on them. And he's got to smell it and then write down which code matched which one. Once we start the real panel, they will sit in the booth. They will go into a booth, set of reference standards, and they would have to mark each, match them all up. They get instantaneous feedback, whether they had them right or wrong. And if they had them wrong, they've got to go back and redo the ones they had wrong. We just actually created a little, if they do get them right, they now get a little clap, clap, clap to say, yes, that was good. Once they've done that, they will then go to the next booth, where they are going to then do the rating for each wine, for each attribute, in triplicate over, in this case, 6 weeks. At the end of which they know pretty much everything there's to know about these wines. So, reference standards. Reference standards can be actual products, they can be compounds, they can be verbal descriptors. Ideally, actual compounds are the nicest, but they usually don't give you what you're looking for. Secondarily, in my lab, we eat food. We are not allowed to have chemicals in the lab. So we very, very rarely use actual compounds. And when we do, we have to make sure that they're food grade. We usually use products. Once in a while, verbal descriptors. So Here, this would have been in wine glasses, not in cups, but for the picture that makes more sense. So we have a veggie here, and it has some green beans and bell pepper in it, because that's what that panel decided. This fruity has some oranges in it, and the nutty has some nuts with no wine in it. Verbal descriptions are not ideal, but sometimes you're stuck with a verbal description. When I was here, we did chocolate milks from the Pacific Rim. We had an Indian chocolate milk that was fecal in odor. It was perfectly healthy to drink, but it stank. The panel leader, to the Trowbridge Center and scraped up some stuff off the floor, brought it back. The panel all smelled and agreed that this smelled like cow shit. And then they decided as a group they really didn't need to do that on a daily basis. And so in that case, we used verbal descriptions. Now, the first and foremost reason for having reference standards is to anchor the concepts for the panel. So everybody understands when they're saying bell peppers, this is what it is. When they're saying sweet, this is what it is. The second reason for reference standards, and the one that frequently gets lost in the shuffle, is it's a translation device. My panel could have called the stuff off the floor Trowbridge, if they wanted to. They would have all understood what it meant. The rest of the world would probably have gone, huh? But if I had explained what it was, they could have gone and found it themselves, right? Here's an example. This is a study on New Zealand Sauvignon Blancs. The student, Cynthia Lund, was a PhD student in Auckland, New Zealand, and I was her sensory advisor for this project. So she sent me, bright and early one morning, her list of attributes. No references, just the list. Sweet, sweaty passion fruit, yeah, that's right. Capsicum, that's bell pepper, I can handle that. Cat urine, boxwood, I can handle that. Passion fruit skin, yep. Flinty mineral, yeah, that's kind of a good, interesting version, but I can handle that. Bourbon. I've drunk a lot of New Zealand Sauvignon Blancs, and bourbon would not come to mind. So I sent an email to Cynthia saying, I need the references, because bourbon makes no sense. So she sent me the references, and that compound is part of freshly mowed grass. Grassy Sauvignon Blancs, those I can live with. Her panel used bourbon as the word. They could just as easily have used grassy. But for reasons that were unique in their situation, I wasn't there. I don't know why they chose that word, but I can translate it. Okay, so let's look a little bit at some data. Let's go back to Cynthia's New Zealand Sauv Blancs and the rest of the world Sauv Blancs. She had 52 wines, which they did in triplicate, from 6 countries. They were on the lower end of the price scale, $6 to $20. Most of them had no oak. They all were at least 90% Sauvignon Blanc. Since they were commercial products, you don't actually know what the total concentrations are because people don't tell you those things. She had 14 judges, and they did things in triplicate. And they came from various parts of New Zealand, the most important being the Marlborough area, 16 of them. Wairarapa, 7, Hawke's Bay, another 7, and then France. Spain, only 2, which became a problem. Australia, the USA, and South Africa. Now, looking at 52 wines times, I think she had 14 attributes, is like Anne Noble used to say, looking at your taxes. It's nearly impossible to see trends. So we've got to take all that stuff and boil it down into a picture. Humans are pretty good at pictures. And if we do that, we get this two-dimensional picture. For most of you, just look at it as a map. In the horizontal, we explain about 43% of everything that had gone on among those 52 wines. In the vertical, another 25%. And we could also look at the third dimension, etc. We're not going to for the purpose right now, but things that are over here, wines that end up over here are going to be high in the bourbon grassy, the flinty. Things down here are going to be more boxwood cat urine. Things up here are not going to be boxwood cat urine, but they are going to be apple candy, apple lolly. Things in this direction are going to be in that more passion fruit stuff. Fresh asparagus sort of is very highly related to boxwood cat urine. passion fruit skin and passion fruit itself is highly related to each other. And then we have a whole bunch of fruity stuff happening over here. So wines over here would be very fruity, but they would not be very canned asparagus-y. Wines in the middle would sort of be a balance of all of these things, probably pretty blah. Then we placed the wines in there. So we had the grassies in the far end, the cat pea over here, the fruity over here, and the passion fruit over here. We go to the next one. Because we had multiple samples, we can get a confidence interval. We can see how spread out those things are. We also had a lot of Marlboros. So Marlboro was not very spread out, and because we had a lot, the confidence interval is even tighter than it would have been. The Wairarapas, which is on the south end of the North Island, sometimes very much resemble Marlboros, but other types probably don't. If we look at the Hawke's Bay, this little grouping here, there's not very many of them, but they're very, very similar to each other. They are also, as it turns out, quite similar to the French, the Australian ones, or the USA ones. The Australian ones intersect with the USA ones, and then the South African ones sort of have an intersect with all of the ones over here. And then Spain, because there was only two, you don't get a confidence interval. Spain, one of them was down here, and one of them was up there. So we can tell something about these wines. We can actually make a claim that Marlborough Sauvignon Blancs are probably different from everybody else's, with the exception sometimes of Wairarapas. And they sell them in that way. Okay, another one, same story. Let's go over this again. This one is a project I did in Australia on sabbatical last year. My brief was to look and see whether there were different styles in low-end Chardonnays in Australia. Low-end, under $15. I was really worried because in the US context there really isn't. So I insisted on having a few higher-end ones. As it turns out, I didn't need them. They were also had to have wines from cooler climates within Australia. And we ended up picking these, so we had the big boys in there. So out of these 21 wines, we had 26% of the sales value, we had 27% of the sales volume, and 12 of all the top 50. So we had a good representation of lower-end Australian Chardonnays. 12 judges, 3 replications, and the attributes. Again, I'm not going to go through these, but what I do want to show is my panel used the word confectionery. When they first said confectionery to me, I had a vision of pastry shops. They had a vision of musk sticks and Allen frogs. I then went, I said, what is confectionery? Oh, musk sticks and Allen frogs. And I'm like, what the heck are those? So off I went to Woolworths, wandered up and down the aisles, finally found musk sticks and Allen frogs. The combination to me smells floral. And so for the publication, I'm using the word floral rather than confectionery, just because it just doesn't make much sense to an American. And then we had a whole bunch of other things. We had a huge argument about the melon. It finally ended up being a honeydew melon without wine to get it to fit their requirements. Again, same picture. This time, 50% in the horizontal, 11% in the vertical. We have sort of this green apple tropical fruit thing going on here. We have apricot peach. We've got sour fresh veg and astringent. And we've got oaky char, honey, butter. So these are the things that saw a lot of oak. These are the things probably that didn't see a lot of oak. And then we've got this creamy mouthfeel up there. It's really thick mouthfeel. And then we have some things down here that are less pleasant. In this case, my 95% confidence intervals are these balls. And really all I want to show you here is these two here are unoaked, or unwooded as the Australians call them. And they are the most fruity. Didn't have the oak to cover up all that stuff. The stuff over here are much more oaky. We've got a few down here that are a little sour. And we have one little guy up here, W5, which is in that creamy mouthfeel situation, this one here. This is actually the second time I've had this particular wine in a panel. I did it in an American panel looking at Chardonnays from around the world. And that guy there showed up as the creamy mouthfeel king. This is Yellowtail. This one here is the largest selling unwooded wine in Australia. Its brother is very similar, yet for one, you're going to pay this one, you're going to pay $3.50 if it's not on sale. All of the ones I bought for the panel were under $2. This one here, you're going to pay about $14, and it doesn't go on sale. So if you like this kind of wine, that's the one you want to go for. We also over here have a couple of wines playing outside of their comfort zone. These are the very expensive ones. In actual fact, this one here was $47 a bottle. These guys are under $10 for this one, and they're not significantly different from each other. If I like that kind of wine, I'm not going to be spending here. I'm going to be spending up there. Last one, 2 more minutes. We also clustered them, and sure enough, there's definitely different hierarchies, and I would actually have put number 5 in its own little cluster, but my co-author decided that he would prefer not to. Australian Cabernets. There's an Australian theme here because this was a doctoral student in my laboratory who came from Australia. He was interested in seeing whether you could tell differences in regionality between different areas of Australia. So they call them geographical indicators. We had 10 of them, 3 wines from each. He went to the wineries and said, out of your 2009 wines, pick the barrel that is the most typical and representative of that you produce, then bottle it and send it to me. So they're all 100% cab, directly bottled, 2009 vintage, bottled in August 2009. We tasted them in March of 2010, 18 judges in triplicate. So they came mostly from the cab areas of Australia. 3 of them came from Western Australia. The other 7 came from right on top of each other in some cases in Southern Australia. But they were all in different categories. And we were interested to see whether you could tell them apart from a sensory point of view. We also did chemistry and all the rest of it, but that's not the point today. Tony's reference standards. He had this eucalyptus one, this earthy one, in which we actually used portobello mushroom ground, the stuff that you're going to grow them in. And this one, we need a 3-dimensional look. So we're going to look at the first 2 dimensions and then look at the 3rd dimension. First dimension, and I didn't put all of them on here because it got very messy. First dimension, about 50-some% in the horizontal, another 21 in the vertical. We have bell peppers and berries. We have astringency, bitter, smoky, and canned veg as sort of the drivers in the first dimension. And part of it that bothered me is that these 2 things are not really related. Usually if you have high one, you have low of the other. Well, it turns out if you look in the 3rd dimension, they are separated. So these things are coming out at us and things are going into the board. But let's look at the wines. We have all the wines are separated with 2 exceptions. The Rattenbully and the Coonawarra, which are right next to each other in geographical locations, are not separated from each other in the first 2 dimensions, as are the McLaren Vale and the Langhorne Creek. They're not separated. Everything else is in its own little world. If we, however, look at the 3rd dimension, which was also significant, we have the mint eucalyptus stuff coming out here, and we have this dry floral stuff coming out here. If we now look at the bubbles, Langhorne Creek is up there, McLaren Vale is down here. Langhorne Creek is high in eucalyptus mint, and from a chemical point of view, it's very high in eucalyptol. McLaren Vale is not. And Rattenbully and Coonawarra are also separated from each other in the first and the 3rd dimension. They also separate from each other in the 2nd and the 3rd dimension. So all 10 of these are literally their own little world. And from a sensory point of view, you can tell the difference between them just looking at the wine, no other information. You can also tell the difference looking chemically. Okay, now, none of you are going to run off and go start doing sensory panels. But all of you can run off and make some reference standards and try to see if you can find them in wines. All you need are some glasses, some household stuff, preferably if you're going to do it in a wine, a box wine that is really bland. And you can start throwing things in there and see if you can smell it in the wine. Can you smell it and you can train yourself. You can also go off and buy something that will train you to do this using real compounds. It's horrifyingly expensive, and those compounds tend to change very quickly. You're better off making your own. I teach this class in Davis, and 2 years ago, the last time I taught it before I went on sabbatical, one student came into class about halfway through the quarter and said, "My wife is about ready to divorce me." And I say, "Why?" It's really my problem. He said, well, every night I'm taking absolutely everything in the kitchen and I'm putting it in wine and I'm smelling it. And do you know that cassia smells different from cinnamon? And do you know that there's 5 or 6 different kinds of cardamom you can buy? And I was like, yeah, it's really great that you're interested, but let's work on the marriage first. Any questions?
Share with us the creamy yellowtail.
The creamy yellowtail.
What was the W11 and W12? The expensive versus the cheap?
Neither one of those are available in the United States.
So I was curious about, you've got all these different wines, determine what dimensionality is in the space that you plotted them in, and what exactly.
Multivariate statistics. Okay. In this particular case, I used principal component analysis for the first one and canonical variant analysis for the other 2.
So you talked earlier about the individual variation in olfactory receptors and taste receptors. There's also some evidence for that training actually changes the biology of the system as well.
It can in some cases, and especially there are some compounds where you may not be able to smell them to start with. Diacetyl as an example. And if you get repeated exposure to diacetyl, you eventually will actually be able to smell diacetyl, which smells like buttery. It's a buttery. If you buy butter buds, that's what diacetyl smells like. Great standard, by the way, for oak Chardonnays. But there are other compounds for which you need to have the receptor there. It may not be expressed for it to eventually be trained into. But if you don't have it to start with, it's never going to come. And so there are some compounds for which no matter how much training you do, it's not ever going to improve.
So how do you deal with that in your panel?
Okay, so that's why we have lots of people. By having lots of people, some people will not, say, get diacetyl. And so they score diacetyl low. Other people will be really, really sensitive to diacetyl, and they score it really high. So we end up with sort of an average diacetyl, which is probably close to what the majority actually smells. And that works most of the time. If you are really, really interested in let's say we, for some reason, have to concentrate on bitter. That's really for whatever reason for the question we're answering. Bitter is more important than absolutely anything else. Then we will probably screen them for bitter. But most of the time, we're trying to get snapshots and more generalized. The other thing is if you're not sensitive to something, it doesn't mask other things. So for example, I can't taste caffeine at all. So to me, decaffeinated and caffeinated coffee tastes exactly the same from that perspective. I know what bitter is because I can actually taste amino acid bitter. But coffee is really sour. And many people who taste bitter don't get the sour. And so I'm really sensitive when it comes to sour. I'm not sensitive when I get to bitter. Yes, sir?
You mentioned that the expensive test compounds change with time, and so does wine.
Correct.
You open the bottle or you leave it in the glass.
No, correct.
So forth. How do you deal with that in these kinds of panels?
In the sensory panel, we make sure that everything is poured in a very specific window. So they get, if we need to look at what happens in a wine that you would have wanted to breathe before or decant, we will actually pour them in advance and then serve them at the time we need it to. If we just want to get it out of the bottle directly, we'll serve them as they come. They sit down and we pour. We're in the middle of a sparkling wine panel, and there you absolutely have to do it within about a minute after opening the bottle. We have to serve the panel. So we control, control, control.
And I guess the question is, some of these compounds obviously change with time. So for example, I really don't like the cat pee, a New Zealand Sauvignon. but on the other hand, fine.
It does. It doesn't totally.
drinkable.
Yes, it doesn't totally actually. This is thiol. It's a sulfur compound, so it's extremely volatile. But it's not so volatile that you will lose absolutely all of it. But you make it, in your case, down to below what your threshold is and be happy with it. People who are really sensitive to that, no amount of swirling will remove cat pee.
I've had cats with the same experience.
I know this is about variations in human sensory, but cats, we
Yep.
live with cats. I've noticed cats who's also affected by onions when you're cooking.
They also
I've
have.
seen cats back away from celery and banana like it had toxic fumes. But I've got one cat, you cut a melon, whether watermelon or other cantaloupes, and he rushes to it to lick it and just
I mean, our senses vary. Every animals vary in the same type of way because I think it's only in the last about 15 or 16 years that we're starting really to come to grips with the fact that there is this huge variation. When Amor did his work in the 60s, he was pretty much laughed at. It's like, well, you didn't do it right. Well, he did it right. Hi, Laura.
Hello, Hildegarde. My question for you is, do the two Australians, well, the study in New Zealand and then the Australian one that you do, it seems to support the concept of terroir? Is that,
I don't.
I mean, is that part of what's underlying?
I very strongly hate the word terroir because terroir actually means more than just geology and climate. It also includes culture and all kinds of other things. I very much prefer the word regionality. And yes, those two studies do show that at least for those places there is some strong regionality effects based on the climate and the soil and the growing methods.
So I was really interested in the amount of variation that exists not only in taste but in smell and sight. That was really fascinating to me. Have people done studies that plot whether there's consistency between people who see, smell, and taste in a certain way, and then different people see, smell, and taste in a consistently different way in that group, or are each of those on its own axis?
Each of those is in its own axis, and so they can be multiplicative in the sense that, nobody has actually done a cross-modal type work on this. Interesting that you asked that because a group of colleagues and I have just put in a grant to look at what they are calling, and I'm cringing a little at the name, but human phenotyping. And the idea would be to put people through batteries of sensory tests on all the modalities, as well as other receptors and genes and stuff like that, and see if we can come up with some picture.
What about degrees of sensitivity to taste versus degrees of sensitivity to pain in other parts?
Yes, and again, that has not been directly studied. It does seem, however, that people who experience a great deal of pain as being supertasters tend to also experience more pain in other places, but it's very tentative data.
Thank you.
So Todd railed on Alice Waters. I'm going to bring up the other Alice firing in the whole natural wine craziness. Is there anything to it? Can your testers tell the difference between something that was done by a guy with his horse and his goat and everything picked by hand, and something that was just done?
We haven't done any organic versus or biodynamic versus normal farming. There was some work done in Australia and some work done in Spain, and the differences were not major. Now, if you go into that's in the wine. If you go into the vineyard, especially a biodynamically farmed situation, the vineyard usually looks better. My contention is that the vineyard looks better not because of the cow horns and the phase of the moon and the whatever. The vineyard looks better because the viticulturist is walking through said vineyard every single day instead of driving by once in a blue moon. And similarly, if you live with your children or with your cats, you can tell when they're not well because you see them. But if you only see your cat or your child once in a blue moon, it's probably less likely that you're going to pick up on subtle differences. And so I very much believe it's a paying attention thing rather than a biodynamic thing.
Okay, this sounds like a weird question. It has nothing to do with wine. It has to go back to the boar taint. So when I was a kid, like I was about 14 or 15 years old, for various reasons my family had to butcher a young boar, and we had to eat that meat, which was the worst thing ever. In fact, I couldn't stay in the house when that was cooking. Now my mother could smell it, but my brothers and my dad, what's the big deal? So I'm curious, what was going on?
There's a genetic component to the specific anosmia of boar taint. That 5%, the majority of men cannot smell it. Or smell it and say, yeah, it smells like gym socks. It's not really bad. Majority of women can smell it, and a good chunk of them find it nauseating. So there's a huge difference in that.
What about age? Does that matter?
No, age doesn't make it. It's gender. But boar taint is one that you can induce. So somebody who has never been able to smell boar taint can actually start smelling boar taint if you induce it by repeated exposure. I'm surprised that you still eat pork. My father tells a story of his first job. He was a civil engineer, and he had to go out on a road construction site, and he lived with some old lady who the day he got there slaughtered a boar. And when he left a month later, he had eaten the entire pig. And he's highly sensitive to boar taint. He has not eaten pork since. He was 22, he's 82.
I never ate that meat because I was physically sick from the
smell.
I took one bite one time, and that was it. I never ate any of that meat.
Okay.
I'm interested in how this information is used. For example, do wineries use this to make any changes in their winemaking process or in their marketing, or just how are they interested in it?
Okay. They, all of the above. Frequently this is done by wineries to make changes in their blends, because you can also then take this information and project the consumer information on top of it. And then you can find out, for example, that your competitor, who is selling a lot more than you are, is making, let's say, that $3.50 wine that is unwooded, and yours is really heavily wooded. So if you change that, you probably will sell more. So the wineries use it for that purpose, for blending and marketing purposes. We use it mostly because we want to, frequently we'll do looking at what happens in the vineyard and does that carry through into the wine. Because a big change in the vineyard tends to lead to a smaller change in the grape, a smaller change during the fermentation, and does it even still show up. So if you're going to spend a lot of money in the vineyard and you get this big of a change over here, it's probably not worth it. But if you end up spending a lot of money and the change is this much, it's probably worth it. These three are sort of anomalies in the sense that we really just looked at commercial wine. In the first case, the 17 high-end wines was paid for by a company where actually 11 of them were the very high-end, and then they had 6 wines that were Me Too's. They were much cheaper wines that wanted to play with the big boys, and they wanted to see if they fit into the sensory profile of the big boys. The Chardonnay project is part of a very large Australian project in which they are currently having about 12,000 consumers taste these wines to try and see what the segmentation for Chardonnay in the Australian wine market is, because the consumption of Chardonnay in the Australian wine market has done this, and they're trying to find a way of bringing it back up by figuring out what people like. The third one was, my student was a flavor chemist and had a great tool chemically to separate all these wines, but he wanted to see if all those differences actually translated into the final product. And that's why the Cabernet. Dean Payne.
Comment and two questions. First comment is, instead of driving you around, Dave was just turning the gas jet on.
Okay. Doesn't smell the same.
I know. I remember you telling me that skunk is in our wine.
Yeah.
First question is that I've always been
wondered what a technological example would be for why we breathe one type of wine and we tend not to breathe the other.
Mm-hmm.
Why we chill one type of wine
Mm-hmm.
and we don't chill the other, which is probably
Mm-hmm.
the same base. What's a very quick...
Well, the temperature situation is if you have something that's cold, you have less volatiles, and therefore it smells less. For example, if you are sensitive to port taint, eat your port cold. It helps. And also swallow it without chewing, that helps. So that's why. In the case of when we do the studies, everything is at room temperature, because we want as much flavor to come out of that glass as we could possibly manage. In the case of white wines, we traditionally serve them cold. We tend in this country to serve them too cold. We're usually at refrigerator temperature, 40 degrees or so. We should be at about 50. And traditionally, we serve red wines warm, hot, room temperature, and we serve them too hot. They should actually be, think British castle room temperature, 60, 65 degrees, not whatever we usually serve them at.
So the compounds in those types of wines,
changes.
whites versus the
There's a
reds,
huge change. Yep.
appreciated at
Those temperatures, yeah.
Corks versus
Corks.
caps.
Screw caps.
Yeah.
You want me to get on a...
Yeah, I talked about this before, but the New Zealanders and the Australians, of course, are screwing with screw caps. And, but I was in a conversation last week over there on that very subject, and we were talking about some Pinot Noirs which they are producing on the South Island, Pyramid Winery. I don't know if you know the factory that he's from the States. But the comment was made that yes, it is important to have wines receive something through a cork, which they can't
Okay.
through a cap, and that's always
Well, they can actually get it through a cap too these days. What he's talking about is if you make a wine the old-fashioned way and you stick it in a screw cap, especially white wines and especially Pinot Noirs, they're very reductive. There's no oxygen there. You've got a very reduced sort of environment, and you tend to showcase the sulfur compounds, rotten eggs and things like that. So what Australian winemakers have been doing, because the New Zealanders, is you make the wine slightly more oxidative. In other words, you are not quite as anal about keeping the air out. You actually allow it to get a little bit of air, usually in the bottler. Instead of pumping it into the bottom of the bottling container, you actually let it drop through the air for a few inches, and that picks up some air, which will then go into the bottle, and you'll end up with a non-reductive wine. But you need to know what you're doing. So now you can actually buy screw caps that have microscopic holes in them that let through minuscule amounts of oxygen, the same way a cork does. Now, I don't understand what American consumers are thinking. If you do a study in Australia, New Zealand, the UK, screw caps are better than corks. Wouldn't you want to drink a wine that the winemaker bottled, put a seal on, and you get it exactly the way he or she envisioned it to be, versus a bottle where the winemaker stuck a sealer on it and is praying that this thing would not have cork taint in it? The problem is everybody else sees this as a perfectly good thing, except us, the Portuguese, and the Spaniards. And they grow cork. What's our excuse?
I like screw caps.
It's so much easier. It's so much easier, much less hassle to deal with, and the wine is usually a lot better. We did some work when I first went to Davis, in which we looked at a very high-end Napa Valley Cabernet. When we did this was 2005. The wine had been bottled in 2006. Some of it had been bottled in cork, some of it had been bottled in the old screw caps. And when we tasted this wine in 2005, 9 years old, this is $150, $190 a bottle. The corks were dead. The screw caps tasted like they'd been bottled the last week. They were wonderful. Yes, sir?
Good question. How do you select your judges?
How do you
incentivize them to do a good job? And
Okay.
It seems like they spend a lot of time with you for several weeks. So.
Okay, we select by enthusiasm and motivation. We want them to want to be there. We very rarely select for any other reason than that. They are volunteers because we have to go through the IRB. So we feed them. We make cookies, and we make lasagna, and usually at the end of the panel, we will have a big lunch, and we'll tell them what they had just done. We tended to use, until fairly recently, mostly graduate students, because they had the free time, in quotes, and they were over 21, because we had that problem as well. Lately, we seem to be using mostly women, but somewhat men of what I would call a certain age. These are people who are retired, and most of them, it started with one of our panelists' mother was in the Master Swimmers in the Davis campus, and so she volunteered, and then more of them volunteered, and more of them volunteered. And so at this point, we have a group of people who are, like Dr. Manning, who are retired, who love to feel needed, who really like what they're doing, and they're really willing to come and do it. In the worst case scenarios, when we have something really horrific for them to taste, we can usually get away with a $10 gift certificate, but getting that through the IRB is not worth it. So it's a lot easier to just go with lots of fuss and bother and cookies.
A quick follow-up. Is there any correlation between the results from these kinds of non-expert panels and some expert judgment or sensory analysis on these wines?
Expert.
So in other words, why not get an expert?
The short answer is no, mostly because the experts are not reproducible. There's a lot of publications right now out, Hodgin, Gall, a few others, looking at Australian expert wine judges. These are people who do wine judging for the gold medals and stuff they love to stick on their bottles. And the same person will not give the same score. There's about I think 1 in every 50 will do that. So my panel is much more reproducible than that. I mean, in my panel, the interesting thing is we just had a study. We finished a study, and then some other wine came in, and it was like, oh gosh, we've got to get this done. It's in the same IRB requirement. We didn't have time to train a panel. And so I told the guy, this is going to be an informal tasting. The panel is going to had been trained on something else. They're going to do these 6 wines in triplicate, but I'm not putting a seal on these data. We're just looking at it. So we actually had 3 wines in which they had 2 fermentations. So we had 6 wines, 3 times 2, and then they did that in triplicate. So my people of a certain age came in, and they started doing their panel. And Mary, one of them, came down and said, did you give us the same wine more than once this morning? And I said, no, we didn't do that. And then somebody else came and asked us the same question. And these wines were extremely subtly different. What they were is that they were fermented with, one was fermented with a Saccharomyces strain. The other one had 1 day on a non-Saccharomyces strain, and then the rest on Saccharomyces. And the third one had 2 days on a non-Saccharomyces strain, and then the rest on Saccharomyces. So subtle differences. And they all picked up on the fact that they had pairs.
Well, we have time for one more question.
I think I'm the right age. I'm over 21. And
You're welcome. Please move to Davis.
the sign-up booth that we wear.
Yeah.
A quick question, and that is, how do you explain eucalyptus chemistry in wine?
There's a lot of arguing about that one. And the most probable answer to that one is that it comes from the eucalyptus tree, not from the wine. That it gets sprayed into the wax layer on the grape. Because in general, you get more eucalyptol if the vineyard is on the wind prevalent side of a eucalyptus grove. Now, there is some argument by the Australians that it is inherent, but I'm not totally sure of that. I think it's basically just coming from the tree. Okay, thank you.
Thank you.